Hello, greetings. Welcome back to Water Quality. Before we get into today's lecture, I want you to take note of this. We are going to have test number 2, and test number 2 is going to be on Monday. It's going to cover water hardness to a little bit of topic 3, so whatever we cover on Thursday's lecture, a little bit on water biology. So back to alkalinity. So alkalinity is defined as the capacity to neutralize acids, so it's the opposite of being acidic. You're probably familiar with sodium hydroxide, not usually found naturally. So most of our alkalinity is going to come from carbonate, and in many cases limestone, so calcium carbonate. So in this formula here, it says total alkalinity is going to be bicarbonate plus carbonate plus hydroxide and minus protons. But usually in most natural settings, we're looking at carbonate and bicarbonate. And so the units for alkalinity are expressed as milligrams per liter calcium carbonate. So where have we seen this before? Of course, we have seen this with water hardness. And this is, like I said, goes back to kind of, there's some historical reasons for having this, but you know, it used to be assumed that alkalinity and water hardness was from calcium carbonate. Of course, there are other sources of bicarbonate, carbonate, calcium, magnesium, but the units have stuck, so we are stuck with it, and we have to use this milligrams per liter of calcium carbonate. So here are some sample calculations. So how do we figure out alkalinity? So let's write down some steps first. So calculating alkalinity. So it is very similar to figuring out the way that we calculate hardness, except for in this case, we're dealing with carbonate and bicarbonate. So step number 1 is we convert our carbonate, so CO3 to negative, and bicarbonate, convert those concentrations to milliequivalent per liter. Okay, step number 2 is we add them up. And step number 3, we're going to take these and we're going to convert to milligrams per liter calcium carbonate. So let's do this for the first one. Okay, so for the first one, step number 1, we convert the, so we'll do this for carbonate, so CO3 to negative. And so we're going to take our 12 milligrams per liter. We are going to divide it by the equivalent weight, which is 30, I believe. Yeah, so 30 milligrams per milliequivalent. And that is 0.4 milliequivalent per liter. Our bicarbonate, HCO3 negative, it's a similar calculation. We have 100 milligrams per liter. Divide that by the equivalent weight, which is 61 milligrams per milliequivalent. And grab my calculator. Can't quite do that one in my head. Get 1.64. So 1.64 milliequivalent per liter. Okay, so now I'm going to add these up. So 0.4 plus 1.64 equals 2.04 milliequivalent per liter of hardness. So that was step 1. This was step 2. And step number 3 is we're going to convert that to milligrams per liter calcium carbonate. So we take 2.04 milliequivalents per liter. The equivalent weight for calcium carbonate is 50, and that's 50 milligrams per milliequivalent. Scroll down a little bit more, and my answer is 102 milligrams per liter calcium carbonate of alkalinity. Okay, so hopefully that's pretty straightforward. I will do number 2, and I'm going to do number 2 right here, and I'm going to do it all in one step. So big bracket, carbonate is 6 divided by its equivalent weight, which is 30. We're going to add that to our bicarbonate, which is 45, divided by the equivalent weight, which is 61. Multiply that whole thing by 50. Punch that into my calculator, and I get 46.9 milligrams per liter calcium carbonate. So do make sure you take a look at the sample problems that come with these notes. And if you're having a hard time with this, please come talk to me, and we'll make sure that we can help you with these calculations. So there is kind of a balance, right? You know, people want neutral water. They don't want scale-forming water. They don't want corrosive water. So here's a little chart to show you kind of how that falls in when you relate things to pH and alkalinity. So I want to talk briefly about acid-base titrations. We are not going to do them on the tests in this class, but I'll bring you through it quickly. This is kind of chemistry 101 stuff, but a titration, an acid-base titration anyway, is where you're measuring the amount of acid or you're measuring the amount of base that's found in solution. And so in the burette, you're going to have the opposite of that. So for example, if you have an unknown acid, you're going to have a base in the burette, and that base is probably going to be sodium hydroxide. If you have an unknown base, you're probably going to have hydrochloric acid or sulfuric acid in the burette. So it's important to know at least the terminology for acid-base titrations. So how does this work? Well, you're going to have your acid solution. This will be my unknown acid solution, and you're going to quantitatively measure volume into a flask. In the burette, you're probably going to have sodium hydroxide. And then you add your sodium hydroxide until it reaches an endpoint, so you can measure that endpoint with a colored indicator or using a pH meter. And then you read the final volume. And once you have your volume and your concentration of your sodium hydroxide, you know that that's going to react with the acid, probably in a 1-to-1 ratio, and then you can figure out the number of moles of acid in the final solution. So if you don't know how to do this and you want to know how to do this, let me know, but it is not going to be on the test. But like I said, it's good to know the kind of language around titrations. So I'm just going to move through this, and there's some sample calculations. You can try these if you like, but like I said, I will not have one of these samples on the test. I'm going to focus on the other stuff that we've talked about. You can see the idea how it works. Just keep in mind, if you do number 2, we're looking at sulfuric acid, and because that has an H2, it's going to react twice. Okay, so some quick quiz questions here. And you can see I have the answers labeled because I'm not animating this presentation. But you can see it says number 1, blank is the measure of how much acid can be added to a liquid. So if you're adding acid to a liquid, it must be alkaline, and therefore alkalinity is the answer. Alkalinity is reported as, and remember those units, milligrams per liter of calcium carbonate. Question 3, the pH of a solution is the concentration of protons or hydrogen ions. Minerals that can cause an increase in alkalinity. So remember, we're looking for carbonate and we're looking for bicarbonate. So those are the ones. Those other things, sulfates and bisulfates and chlorides, yes, they are conjugate bases, but they are very, very, very weak. And so they're not going to be significant. So let's switch gears for a few minutes and let's talk about other things that can be dissolved into water. So ions are obviously very important. Some of them are reactive, some of them are causing alkalinity or hardness and those kind of things. But there are quite a number of other things that we can be concerned about that are dissolved in water. So let's take a look at some of these gases. So first one here is methane. And methane is basically from decomposition. So we're talking about you can see methane gas in a lot of swamps. Not very soluble methane in that we can assume that zero methane is actually dissolved in water. O2, of course, is oxygen. So oxygen is going to come from photosynthesis, and it's also going to come from the atmosphere. So you can think of waves, of course, are stirring stirring oxygen into the water, right? NH3 is ammonia, and this basically comes from, it's a waste product, mostly fish, but other animals. Basically, it's in their urine. So this is the kind of thing that if you have a fish tank at home, you don't want to have too many fish into one fish tank, or they will actually poison themselves with too much ammonia. So H2S is, of course, hydrogen sulfide. And this is also from decomposition. It's a pretty toxic gas, so I think next year you're going to take that safety course, H2S Alive, and learn all about how to protect yourself from hydrogen sulfide. CO2 is carbon dioxide. So this is from respiration and burning of fossil fuels. And the last one, of course, is chlorine. And this is added by humans as a disinfectant. So out of all of these, one of the ones that we're concerned the most about is oxygen because it's important for wildlife and fish. So we'll talk about that in a moment. So before we get to oxygen, one thing to consider is how much gas is in a solution is actually related to pressure. So this is something called Henry's law. Don't worry about this formula. What I want you to think about is what this formula says though. So this is the amount of gas in solution and it's related to the amount of pressure. So what that means is that the amount of gas in a solution is directly proportional to the amount of that gas that's in the atmosphere around that water. Okay, so let's talk about dissolved oxygen and talk about some implications of that. So first thing to know about dissolved oxygen is that as the temperature decreases, so notice the temperature is decreasing, the amount of oxygen increases. So the implication of this is that some fish in the summertime when it's warm, so I'm thinking about something like lake trout, they go very, very deep because that's where there's more oxygen. And in the wintertime, they're not deep. And you can ice fish for them and very easily because they're a lot closer to the surface because there's a lot more oxygen up there. They're probably also foraging for food as well. But a big part of that is lake trout. Lake trout tend to be quite sensitive to low oxygen levels, by the way. So oxygen dissolves greatly with decreased temperature. We talked about that. It turns out that salt, in fact, will affect the solubility of oxygen. So you can see that it says here gas solubility decreases with increased salinity. That means salt water has less oxygen in it at equivalent temperature and elevation and those kind of things. Oxygen solubility varies a little bit with atmospheric pressure. So that also means if you climb a mountain where there's less pressure, there's going to be a little less oxygen in the air and therefore a little bit less oxygen in the water. Of course, it gets colder up there, which helps increase the solubility of water. So it kind of cancels out a little bit with mountain elevation. So why is dissolved oxygen important? Well, because of fish. And of course, you may not care about fish yourself, but lots of people do. And of course, lakes and rivers and ponds that have fish in them tend to be a good indicator that you have actually healthy and good pristine water. The more oxygen usually is an indication that the water is better quality. So this is something that we definitely pay attention to is dissolved oxygen. So by the way, something else to consider is that oxygen does turn over quite a bit in lakes in the spring and fall with overturn. So if you don't know what that is, this is where the tops and the bottoms of lakes mix with the seasons. So as the temperature cools and heats, the density of water changes, and this causes a lot of mixing in lakes. And so you've got nutrients from the bottom brought up and things like that. And so this will affect the dissolved oxygen levels as well. And last thing to think about is the weather, right? Different things can happen with the weather. Sunlight, of course, is heat, and heat will decrease the amount of oxygen in a lake. If you have waves, think about a still pond versus a bubbling brook, and the water is flowing over rocks, and that's adding a lot of oxygen to it, to the water. So all these things can affect dissolved oxygen, of course. So one thing to note about dissolved oxygen is that it's not that soluble in water. Oxygen is actually a non-polar molecule, and water is polar, so they don't mix very well. You can see if you take a look, a liter of air is about 280 milligrams of oxygen, whereas a liter of fresh water, if you've got pristine water, maybe around 15 milligrams of oxygen. If you have less pristine water, it's going to be less than that. And so, you know, that's not very much, but it's enough for most fish. 15 milligrams per liter of oxygen is actually enough for most fish, and they're very, very happy with that. Here are some numbers. You can see this is talking about as the dissolved oxygen in milligrams per liter kind of decreases. You can see that once it gets down to 6 for most species, that's a crucial level. Any lower than that, they can't spawn and lay eggs and whatnot. So this is something, like I said, it's going to be obviously species specific. Some species are going to need a lot more oxygen. Some species are going to be fine with a lot less, but kind of gives you some guidelines. So last week's lab, you had a chance to use a DO meter, and the ones that we have in the lab kind of look like this. And one thing to know about DO meters is that they consume oxygen at the tip of the probe. And so usually you have to stir them a little bit. So the ones we have in the lab have a little button on them, maybe right there. And when you put it in the sample, you turn on the button and it stirs the water in the proximity of the probe. I've used a type that looks something like this as well. And these probes, you have to stir it yourself. So you may stand near the stream and just sort of move the probe around slowly. Because if you leave it in one place, there's a dead zone of oxygen around the probe, and it's going to read incorrectly. Usually these are pretty finicky, and they don't necessarily stabilize very well. But you know, usually you can get a ballpark number. You know, you can know if it's 3 ppm, that's bad. If it's 10, that's pretty good. If it's 15, it's a lot better. I'm assuming this one here is also a field meter that you have to stir by yourself as well. So this is a question that comes up sometimes. Sometimes you turn on your tap, get a glass of water, and it looks all cloudy. And this is actually nothing to be worried about. This is just a lot of taps now have little filters in the nozzles, and it puts a lot of air bubbles in there. And this is just completely harmless. If you leave the water for about a minute or two, usually those settle out and you don't see them anymore. So just a little interesting tidbit for you. So what else can end up in water? So something called colloids are something that can end up in water. So these are kind of larger particles or other things. And I wouldn't necessarily call them molecules. Usually they're kind of clumps of molecules and whatnot. You can see some of the things this might include. A big one, of course, is clay particles. If you live around Fort McMurray, there's a lot of clay in the river. And of course, microbes and decaying matter and things like that. A lot of these things will affect the turbidity of the water because the light can't penetrate, and so it might make the water look cloudy in at least small concentrations. Most of these things don't necessarily, they might not necessarily be bad for human health, but of course, if you have water coming out of the tap that's colored, people are not very happy with that. So it's something that does have to be removed for water treatment. So a lot of these colloids, and particularly clay particles, don't settle out very easily because they all have similar charges. And so they have negative charges, and negative charges repel negative charges, of course, and so they don't settle out very well. And so there's a process for removing that. It's called coagulation. We're going to talk about coagulation when we get to water treatment, and that will be a little bit later on in the semester. So another thing that can end up in the water are organic compounds. And so by organic compounds, we mean compounds that have carbon and hydrogen. And we're kind of talking about all the 4 food groups. And by that, I mean, you know, kind of your usual biological molecules. So we're talking about proteins and we're talking about lipids, kind of are the main ones, but you can also talk about carbohydrates. We could talk about nucleic acids and many synthetic and otherwise compounds. And so I'll give you some examples in a minute. I'm going to skip this thing here. You can savor that and wait for organic chemistry to talk about constitutional isomers, but I'm going to skip that for now. So let's talk about some of these things. A lot of these products are just kind of natural, found in water sources. You could think about animal feces or just decay of plant and wildlife, et cetera, et cetera, et cetera. Some of these things are from humans. So you may or may not have heard of naphthenic acids. So naphthenic acids are actually a byproduct of bitumen extraction. And those tailings ponds that everyone talks about are loaded with those naphthenic acids. Some of them are really stable compounds that don't break down very quickly. So this is one thing that we're very, very concerned about. A bunch of other things, if you think about Fort McMurray and how many people on a daily basis are having, let's say they have a Tylenol, that's acetaminophen, having a cup of coffee, that has caffeine in it, treating an infection with antibiotics, or women on birth control. So all of those things, your body absorbs some and the rest will go out through your urine. And so on a daily basis, we're actually releasing quite a few pharmaceuticals into our wastewater. And there is some debate about how much that is affecting the wildlife. There are some good studies showing estrogens feminizing fish. I don't know exactly what feminizing means, whether it means they don't want to mate with the females or whether it's actually affecting their sperm. But there are some good studies to show that that is the case. But those are at high concentrations. I think the debate is whether the amount that we are releasing, how much an effect, if any, it is having on the wildlife. But anyway, something you can look into for other courses. Of course, disinfectants and pesticides, around farmland, there's pesticides that are going into the waters as well. So these are things that we're concerned about, of course, that are all fall under the category of organic molecules. So I'm going to skip this for now. We're going to come back and talk about measuring organic matter in topic 3. So more on that later. What I do want to talk about are these important water quality parameters. So a bunch of these we've already talked about already, right? Dissolved ions, hardness, pH, acidity, alkalinity. We're going to talk about turbidity a little bit later. But you can see that some of these other things, I just want to kind of talk about total dissolved solids for a few minutes. But all of these things are things that are measured at water plants, and they can affect different processes and whatnot, and affect human health in some cases. So they're all important parameters. I have radioactivity in brackets, by the way, because we don't actually measure it here at our water treatment plants. We don't have any reason to believe our water would be radioactive, but they would measure it if they suspected, let's say there was a spill or something upstream, and then they would of course measure the radioactivity. So total dissolved solids are worth talking about for a few minutes because you're going to see them in this week's lab. And this involves mostly dissolved ions, but there could be other things contributing to it. But most of the usual suspects, right? Sodium and chloride and sulfates and calcium and so on. So some of these may affect the taste of the water and obviously the reactivity. And so this is something that we do want to measure and think about when we're looking at water quality parameters. So there's a little diagram showing different brands of bottled water. And so if you remove all of the solids and ions from your water, you can see what it's showing here is it basically tastes bland. People don't like the taste of that. So there is a sweet spot. It's around 100, 150 ppm of salts in the water. So you can see that's where most of the brand of water are falling. Once you get around 500, the water is going to start to taste salty. So I'm not familiar with this particular brand here. I can't even read it. It's a little bit small. But hey, some people actually kind of prefer that. And Perrier, you can see, is pretty high on this, and it's a pretty flavorful type of bottled water. So just something to think about for that, right? So there are all sorts of things that can add dissolved solids. You can think of all the pollution, of course. We've got, we're using road salt during the wintertime, all the other human processes and industries and and those kind of things can add to the total dissolved solids. So this is something that with wastewater treatment they have to get down, and they're able to control that quite a bit. So what happens if we have too many total dissolved solids in the water? Well, first of all, it might taste bad, right? So you can see there's that number, 500 ppm. And when the water is around that level, it starts to taste salty and people don't really like it. So that is the maximum level that's allowed for drinking water. Sometimes if the water is too high, like I said, it's going to affect industrial processes. And of course, it's not necessarily that good for human health either. So I want to finish off here because this is relevant to this week's lab. And this is how we are going to measure total dissolved solids in lab 3. We are going to use a conductivity meter. So this is what we have for conductivity meters. These are field meters, and you have to calibrate them kind of like the way that you calibrate a pH meters. You have some standards and you calibrate them that way, and then you get a reading. And the reading is going to come out in Siemens per centimeter. So usually it's microsiemens or millisiemens per centimeter. So make sure you're careful with those units, that you take a look at what units you're reading, because I think it switches automatically depending on the actual reading between micro and millisiemens per centimeter. So what is conductivity? Conductivity is the amount of electrical flow that can go through water. And basically, the more ions you have, the more electricity can flow through the water. And so it's directly proportional to total dissolved solids. And in the lab manual, there's a conversion. I can't remember what it is. It's something like 600 microsiemens per centimeter is 1,000 parts per million TDS or something like that. You have to take a look at the conversion in the lab manual. For that. But that's what conductivity is. So like I said, it kind of gives you an estimate of the total dissolved solids in a very quick way, without having to dry down the water sample and measure it in a balance. You can do it very quickly. And so it's a very commonly used water parameter to estimate the total dissolved solids. So that is it for today. We're going to start topic 3 next day, which is water biology, and I will see you then.